I'm glad to share with you our recent establishment of the a big data analysis platform unveils the gene interaction in cancer. So this is a slide showing the cell in the nuclei, majority of the DNA localized in the nuclei. And there's another important compartment called nucleolus, and which is the site of ribosomal biogenesis. The ribosomal RNA processing and assembly, the site of ribosomal biogenesis. So back to the years 1960s, Professor Harry Bush found the difference between the cancer cell and normal cell. The main difference in the cancer cell and the normal cell is the nucleolus. And the nucleolus is being enlarged and also have many different shapes and also hyperactive in ribosomal biogenesis. At that time, people, as MD and PhD students, and they thought that this is the origin of cancers. So they try making a lot of effort to isolate the nucleolus, to understand the biochemistry in the nucleolus. So so they're using the 2D gel electrophoresis to isolate the nucleolus proteins. Okay. So, and they name the proteins by dividing the three areas, A, B, C, and just name 1, 2, 3, 4, 5. And this is B area, protein B23, and also the C area, also C23, are two major proteins they are interested in. The reasons are these two proteins are phosphoprotein and also abundant in cancer as compared to normal. At that time, I was a PhD student. I was assigned my PhD thesis project to study the B23 since my name have a Benjamin. So I got B23 and I start my career studying this protein since then, B23, okay. So this protein getting more famous and famous in these past 30 years. And in about 2006, there's a Nature reviews and discuss about this protein, how this protein could contribute to tumorigenesis, okay? And in this review, a lot of our publications have been cited. I can tell you, since I was independent as a faculty member in Chang'an University in Taiwan, I continue my study on B23 after I got the training from a Department of Biochemistry at Stanford, and I tried to study biochemistry of this protein. So this protein, also called B23 nucleophosphine, NPM, so involved in growth and cell cycle regulations. And also induce differentiation and apoptosis. And also B23 is involved in the control of DNA damage in response, the cellular response to DNA damage, repair, and also regulation of pCNA. And also, very importantly, associated with oncogenes like RAS, c-MYC, p53. A lot of studies showing that interaction between NPM1 and p53 in control of the apoptosis and so on. And also, we find the NPM1, B23 also involved in the transcription regulations. And there's B23 as a novel AP2 alpha binding transcriptional co-repressor during the cellular differentiations. And also, the phosphorylation of B23 by the PP1 beta facilitate RB binding and regulate the E2F1 dependent DNA repairs. So a lot of studies being done on the NPM1 for the last 30 years. So in the recent years, I and my colleagues at PolyU try to establish so-called gene co-expression networks. Co-expression, I try to explain what is the co-expression network in a minute. We try to establish this network to investigate the intergene association by co-expression, intergene association in expression profiles, reflecting the functional linkage. Their co-expression try to reflect the functional linkage and potential coordinate regulations, okay? So what is the co-expression structures? The co-expression structures is defined as the distribution of co-expression levels for a group of genes over stage. And this structural analysis seeks to identify a group of genes whose co-expression structures in one stage, such as neoplastic subjects, compared with the normal stage, significantly different from that in another stage. Let me show you the pictures to explain. Like this, in the normal cell, these three genes are co-expressed together. Gene A co-expressed with B and C, and using the mathematical model to calculate the co-expression index. So D, E, F, G co-express together, and you can compare with the disease. We find out that some genes are falsely connected in the disease. Before they are not connected, but here, D, E, F, G connected with A, B, C. This group of genes co-express together. And some genes are falsely disruptors, compared with the normals. So this is the co-expression network which tries to establish. Now, comparison of analytical method. The traditional method, what we have been trying to do is there are 20,000 genes that we try to compare the gene expression. Okay, microarray do this and try to see how many fold there are differences, and we try to quantify in the expression of individual genes and differential expression of individual genes. But here, our platform, we can have 200 million gene pairs. They co-express together. So we quantify the gene interactions to establish so-called the gene co-expression networks. Okay? Interestingly, we try to do this network genome-wide and also specific gene focus, NPM1 B23, NPM1 focus or NPM1 link connections. As I mentioned, NPM1 is one of the very important biological proteins. And our results show this is really true for NPM1, play a very important role in mediating a lot of functional linkage. Now I'll show you we use the CML as a model to start with. This is about 3 years ago. At that time, there's not much data, microarray data in the public domain. Only we can compare live pairs of patients with their normal samples. So, but these live pairs, we went through some difficulties, but we overcome this obstacle to get our results. So you can see in the home genome analysis, you can see there's a lot of genes being disrupted. The connection, remember the diagram A, B, C, D, E, F. So a lot of genes being disrupted. But on the other hand, the NPM1 link connections are being over-connected or so-called involved compared to tumor-specific. Now, when I run analysis data, during this over-connection, there's high priorities, the functional, a lot of genes being connected in cancer compared to normal. Number one is the ribosomal biogenesis. A lot of ribosomal protein being connected in cancer as compared to normal. So because I've been involved in B23 for many years, so we look at the NPM1 closely about the linkage during the cancer stage. So, and we know that NPM1 B23 respond to signal from MEK kinase, PI3K pathways initiated by oncogenic RAS. There's a lot of publication last 20 years about this protein. So 1 year, P53 is the most publication study on P53. And 1 year, NPM1 B23 is most being studied. And NPM1 respond to signal with this very oncogenic pathways. So we try to quantify and compare the state-specific association of NPM gene expression. From the combined, we know that BCR, ABL, MAP, P1VK, AKT, important pathways in CML, in the leukemia. So what we have been doing, using diffusion, this pathway, then this is the oncogenic pathway from BCR, ABL, so go through the uncontrolled protein synthesis for CML. Very interesting. Using our co-expression network, they can find out the MPN1 is closely linked to these proteins, oncogenic pathways, co-express, as compared to control, MPN1 linked to these two. So MPN1 is significantly co-expressed with gene in the BCL ABL pathway. Okay. So to further understand the MPN1, as we know, this is the nucleolus protein. It's translocated from nucleolus to the nucleoplasm, back and forth. So a lot of studies where MPN1 is involved in the ribosomal biogenesis. So we try to look at this, the role of MPN1 in ribosomal biogenesis. So we analyze the co-expression network of beta-20 associated gene in the molecular signature database as a gene cluster covering most of the ribosomal proteins. Very interesting. In the normal group, MPN1 is loosely linked with the ribosomal machinery. But this is importantly linked with 3 important ribosomal proteins. And these 3 ribosomal proteins have been reported in recent years, the important role of this ribosomal protein interacting with beta-23 and also p53 in control of apoptosis. So our study using bioinformatics, biostatistics, and also biochemistry, really the data have been verified by a lot of the biochemistry study in recent years. Okay? So these 3 ribosomal proteins are directly, importantly, linked to the co-expression link with MPN1 and also turn on the other linkage with other ribosomal protein for the ribosomal biogenesis machinery. We're excited about this result because this is the very important linkage about beta-23 with ribosomal protein in the functional linkage for the ribosomal biogenesis. So the BCL oncogenic pathway and also the ribosomal biogenesis. So how about the transcription regulations? So we try to using the prediction of transcriptional regulatory modules database. We identify the transcription factor concurrently target the MPN link doublets and elucidate the effect on co-expression patterns. So we try to find out the transcription factor, regulate, normal, and CML group. Very interestingly, we found out some hard genes, common genes, transcription factor, make for step 5 to regulate the CML co-expression network and normal group. One interesting phenomenon that we observed, common transcription regulate the normal and also the leukemia, but they are oppositely regulated at worst. One is up-regulated, but this is down-regulated. Very interesting. Common gene involved in normal and leukemia. And this is quite striking. We thought maybe different transcription factor to regulate, but they are common transcription regulate. And interestingly, different directions. So transcription factor target different genes altering network links. Okay. So a summary of research findings, the transcription factor regulate the beta-23 and its co-expressed genes, and also functionally regulate the B-cell ABA pathway, ribosomal biogenesis protein synthesis for the alteration in cell growth, proliferation, and survival. So our significance of our finding is we established a novel structural co-expression network analysis, which enables us to unveil cancer pathogenesis and potential MPN1-oriented treatment strategy in CML. And this analysis discovered novel unregulated patterns of gene networks for understanding cancer biology, identifying new targets for treatment, and all this innovation will contribute to great science after all. And target therapy can become more target because we load the pathway after loading the gene interaction, co-expression, transcription factor, and pathways. And also in our laboratories, we also apply this platform to other disease, other cancer, other disease such as diabetes and cardiovascular disease. Okay. So this, and also I'm happy because I've been in this field for a long time, but I just went back to PolyU 7 years ago, and we formed a very good team, so cross-disciplinary collaborations with bioinformatics, biostatistics, and also myself is a biochemistry and pharmacologist, and we have also physiology, collaborate together, enable our team to unveil cancer pathogenesis with expertise in different fields. Okay. So, our university held a press conference for us for our finding, and they try to understand the difference between our new platform with the traditional method. So they came us that our platform can analyze the interaction of 200 million gene pairs in few days, and traditional method, if we try to understand the gene interaction, 200 million gene, maybe we have to do a lot of experience invisible. Remember, I showed 5 slides about B23 biochemistry. This has been done in 20 years with a lot of my students, postdocs. But here, I think using this platform, we try to understand in a much more efficient way. So the project team members from PolyU, as I say, physiology, clinical genomics, and also we have from other universities in Hong Kong. And also we appreciate very much about the international collaboration from Harvard, Professor Chi Hong Lin and her students, Gowing Yong, and they help us in a lot of the biostatistic analysis. And you see the co-expression, we need a dependent variable, that kind of calculation, and there's a lot of difficulties we have to overcome because only 9 pairs at that time. But now, after a few years, there's a lot of clinical data coming out. So, but at that time, they help us a lot in the biostatistic analysis and also do the simulations at Harvard. Okay, so. And so this is the press conference in universities. These are 3 key members in this. Dr. Lauren Chen, he will present also in this session. And Dr. Wong is clinical genomics. Okay. He had a very good connection with the hospital in Hong Kong. So we got a lot of clinical sample, although we don't have a medical school in our university. Okay. So this is our beautiful campus, although small. So welcome to visit us, Hong Kong Polytechnic University. And this is the logo. VP means it's poly engineering, a lot of discipline. Okay. And this is nighttime. Very beautiful nighttime. And this is the new building. It's the, got the award. This is Innovation Tower. But a lot of faculty doesn't like this building. This is ugly, but Taiwan. Okay, thank you. With the LP, yeah.
Yes. Now, does it require mutations in NPM
in
order to create those interactions, or is it simply a differential expression of the normal?
This is from the microarray data. This should be the wild type, no mutations.
So no mutation.
No mutation in this study. But we are also interested about some leukemias have mutations.
Yeah.
So what is the network from this mutated B23? And also, the mutation plays some part of the leukemia. Some leukemia don't have mutations. So by this way, we will try to understand, for example, the RP interactions and also the cancer oncogenic pathway. Are there any differences between the mutated B23 interactions or the wild type with the wild type B23? This is what we try to understand more.
Oh, I see.
Yeah, yeah.
Once you begin to see the correlation or the association of NPM1 with these diseases, can you carry out a validation step where you'll overexpress NPM artificially?
Yes, yes.
And then begin to see the same changes in the signaling pathways that you see by the correlational analysis?
Actually, this already published in just a week ago in the scientific report from the latest publishers. And then we have a functional experiment. What we do is using the residual is try to induce differentiation. And then at that time, NPM1 is being downregulated. And also we check those ribosomal protein, whether they will change accordingly to NPM1 expressions. And we demonstrate. So this is the assay that we have. And yes, we try to overexpress or downregulate those ribosomal protein and also those interacting partners and also NPM1 in the coming experiments. And also we try to elaborate in more cancers, in different cancers. Yeah. And in different cancers, they're different. Some is overexpressed together, some is disrupted. So very interesting in other cancers. Yeah. Yeah.
Yes.
Question. How do you interpret the co-expression? Do you do it by microarray technology, for instance, mRNA?
It's mRNA co-expression level. And there's a paper in 2005 from Stanford University, the head of the Department of Statistics, Professor Wong, and he calculated two genes co-expression index. And then we're using this method to calculate the index. And then, but we try to, we extend this method to the whole genome, to analysis. So, for example, when I coming to express, then he is also co-expressed. Then we calculate our co-expression index. There's a complicated mathematical models by mathematician. Yeah.
Very interesting.
Yeah, yeah.
Thank you very much.
Thank you, Mitch.
And there's a certificate.
Okay, thank you. Thank you, Robert. Thank you.